Thanks for joining us in another fascinating journey through the world of science here on the Science Channel podcast. If you're enjoying these explorations, please do hit that like button and subscribe. It really helps support the channel. Today, we're diving into something that could, well, fundamentally shift how we understand where our cells actually get their power. You, our listener, you shared some incredibly thought-provoking, really cutting-edge research with us. It re-examines the basic processes fueling life.
Yeah, it's pretty exciting stuff.
So we're going to unpack the scientific paper. It proposes a maybe surprising twist on the long-accepted model of cellular energy metabolism. We'll be highlighting not just the familiar pathways, the ones we all learned, but also some unexpected key players.
Right.
Our mission today, to extract the core insights, clarify these sometimes complex ideas so they really click, and hopefully reveal some aha moments about how your very own cells are keeping you going right now.
Exactly. And for decades, the textbook picture has been, well, relatively clear, hasn't it?
Pretty much. Glucose comes in.
Right. Glucose enters the cell, goes through glycolysis, breaks down into pyruvate, and that pyruvate then heads into the mitochondria, the cell's powerhouse.
So the classic story.
To fuel the Krebs cycle, and then oxidative phosphorylation, or OXPHOS. That's where we thought most of the ATP, the energy currency, comes from. But this new research suggests a much more dynamic, maybe more interconnected system. Something quite different.
Okay, let's start with glycolysis then. The traditional view, it ends with pyruvate, right? Off to the mitochondria it goes.
Okay.
But this paper argues the end product is more likely lactate.
That's the proposal, lactate. Think of it as a subtle but potentially really significant correction. And it's something that seems to have been overlooked or perhaps misinterpreted for, well, the paper says 75 years after the initial doctrine.
Wow, 75 years. So what's the reasoning? Why lactate instead of pyruvate being the main endpoint?
Well, part of it ties into a long-standing puzzle. Within glycolysis itself, there's a specific step where this sugar molecule, fructose 1-bisphosphate, it's a key intermediate, gets split in two by an enzyme called aldolase.
Right.
Now, thermodynamically speaking, just looking at the energy changes, making that split happen in the direction needed for glycolysis, it looks unfavorable. It seems to require more energy input than it readily provides under typical cellular conditions.
So it's like pushing a ball slightly uphill, energetically speaking.
Kind of, yeah. It's been a bit of a question mark for biochemists for a while. How does the cell efficiently push past this step?
And the paper suggests a way around this hurdle.
It does, by suggesting we shouldn't view glycolysis in isolation. Instead, it proposes glycolysis works closely coupled with another pathway.
Ah, okay. Which pathway?
The pentose phosphate pathway, or PPP.
The PPP.
Right.
I know that one branches off early from glucose metabolism.
Precisely. Glucose comes in, gets phosphorylated to glucose 6-phosphate, and right there's a fork in the road. It can either continue down the standard glycolytic path or divert into the PPP.
And the PPP's main jobs are making NADPH, right? And some specific sugars.
Exactly. Producing NADPH, which is vital. It's a key reducing agent for biosynthesis and combating oxidative stress, and also producing 5-carbon sugars like Ribulose 5-phosphate, needed for nucleotides and nucleic acids.
Okay. So how does linking with the PPP solve that energy problem in glycolysis?
Well, the novel concept here is that these two pathways are tightly linked, maybe even in a kind of closed loop.
Yeah.
Because 6-phosphate goes into both. The PPP makes its products.
NADPH and Ribulose 5-phosphate.
Yes. And then this is the key part, that Ribulose 5-phosphate can actually re-enter the glycolytic pathway, but sort of further down, and enzymes can convert it back up the chain almost in reverse towards glucose 6-phosphate again.
Whoa, okay, so it's like a recycling system. The PPP products feed back into glycolysis.
Kind of like that, yes. It allows molecules to cycle through, being constantly transformed and fed back, until the original glucose molecule is completely broken down, primarily into CO2 and lots of NADPH through repeated turns of the PPP cycle.
That's a really different picture than just a linear pathway.
It is. And it implies something radical about energy production, too. Some of that energy generation might actually be happening outside the mitochondria.
Outside? Okay, hold on. So if this loop is happening and maybe energy production is happening elsewhere, what does that mean for the mitochondria's role, especially regarding glucose?
Well, this model suggests that the mitochondria's primary role, at least when it comes to processing fuel, might be more focused on breaking down fat lipids through beta-oxidation.
Right, beta-oxidation, that produces acetyl-CoA.
Exactly, producing acetyl-CoA, which is then used in the Krebs cycle within the mitochondria or exported for other things. So in this view, Generating ATP directly from glucose breakdown products might be more of a secondary function for the mitochondria themselves.
That certainly makes you rethink the whole energy budget. We're always taught glycolysis gives a net yield of just 2 ATP molecules per glucose, which, when you think about all the work cells do, it doesn't sound like a huge payoff, does it?
It really doesn't, and that's a key point the paper makes. There are even scenarios, depending on which intermediates get pulled off for other cellular jobs.
Like making other molecules.
Yes, exactly, for biosynthesis. For instance, if an intermediate like 1,4,3-bisphosphoglycerate is converted to 2,3-bisphosphoglycerate, which happens in red blood cells, or if glycerol is produced in yeast, the net ATP from glycolysis can actually drop to zero, or even become negative, meaning it costs energy overall.
So if it's not always about churning out heaps of ATP directly, what's the main point of glycolysis in this revised view?
It seems a major role is providing the building blocks, the carbon skeletons, the precursors needed for biosynthesis.
Ah, okay, like making amino acids, lipids, nucleotides.
Precisely. Especially crucial in rapidly growing cells, like cancer cells, which need to duplicate all their components constantly.
But wait, biosynthesis itself needs a lot of energy, right? Building complex molecules takes ATP.
It does, significant amounts.
So if glycolysis isn't the main direct ATP source, and might even cost ATP sometimes, where does the energy for all that building come from?
Okay, now we get to the really exciting and maybe somewhat controversial part of the paper, this idea of extra-mitochondrial oxidative phosphorylation.
OXPHOS happening outside the mitochondria.
Yes. The paper presents evidence suggesting that the machinery for OXPHOS, the electron transport chain complexes, and the ATP synthase enzyme.
The stuff usually packed into the inner mitochondrial membrane.
Right, that this machinery isn't exclusively located there. It appears to be present and functional elsewhere in the cell, specifically on the membranes of the endoplasmic reticulum, the ER.
The ER. That network involved in protein folding and lipid synthesis, OXPHOS is happening there.
That's the proposal. It's a real potential game changer in how we picture cell geography and function.
Wow. What's the evidence for that? It sounds pretty radical.
Well, it's based on several lines of evidence. One intriguing finding is the discovery of these tiny vesicles, sometimes called mitovesicles, that seem to pinch off from the mitochondria.
Like little packets.
Exactly. And these vesicles appear to carry components of the inner mitochondrial membrane, including the OXPHOS machinery, and transport them to other locations, like the ER membrane or even the plasma membrane at the cell surface.
So the mitochondria are like distributing power packs around the cell.
Sort of, yeah. Or at least the machinery to generate power. There's also evidence from direct contact sites between mitochondria and the ER, called MAMs, Mitochondria-associated membranes, where transfer might happen more directly. And studies showing the ER has some mitochondrial-like properties and proteins.
Okay, so if OXPHOS can happen on the ER, how is it fueled? Does pyruvate go there? Or lactate?
This is where the PPP connection becomes really critical again. Remember, the PPP makes a lot of NADPH.
Yes, the reducing agent.
It seems that NADPH is a key player here. There's an enzyme called transhydrogenase, which is known to exist on the ER membrane.
Transhydrogenase.
This enzyme can effectively convert the reducing power of NADPH into NADH. And NADH is the classic fuel that feeds electrons into complex I of the electron transport chain.
Ah, I see. So the PPP makes NADPH in the cytoplasm. Transhydrogenase on the ER converts it to NADH, and that NADH then fuels OXPHOS right there on the ER membrane, producing ATP locally.
That's the proposed mechanism. It elegantly links the PPP's output directly to ATP production outside the mitochondria, potentially supplying energy right where biosynthetic processes are happening, like on the ER surface.
And this might also explain something. Didn't you mention a paradox with a glucose analog, 2DG?
Yes, the 2-deoxyglucose paradox. 2DG is modified glucose. It gets taken up by cells and phosphorylated like glucose, but then it gets stuck. It can't proceed down the standard glycolytic pathway past the very early step.
Okay, so you'd expect it to block energy production from glucose metabolism.
You would, but the paradox is that sometimes cells treated with 2DG, particularly certain types like neurons or cancer cells, can actually produce more ATP than control cells fed normal glucose.
More ATP. How is that possible if it blocks glycolysis?
The paper suggests it's because 2DG can be processed by the pentose phosphate pathway. Specifically, it seems to be a good substrate for an enzyme variant, potentially driving the PPP on the ER.
Wait, there's a different PPP enzyme involved now?
Potentially, yes. The standard view is that the first step of the PPP in the cytosol is catalyzed by glucose 6-phosphate dehydrogenase, or G6PD. But there's another related enzyme called hexose 6-phosphate dehydrogenase, H6PD, which is located on the ER membrane.
H6PD on the ER.
Oh, yeah, and H6PD has some interesting properties.
Mm-hmm.
It has broader substrate specificity. It can use other sugars like lactose 6-phosphate. And crucially, it seems to have a higher affinity for the phosphorylated form of 2DG than G6PD does.
So 2DG gets into the cell, gets phosphorylated, can't do glycolysis, but H6PD on the ER grabs it and shunts it into the PPP cycle right there.
That's the hypothesis. And because it feeds into the PPP, it generates NADPH, which, via transhydrogenase, fuels OXPHOS, leading to ATP production. This could explain why 2DG allows for ATP generation potentially even more efficiently than glucose going through the conventional, maybe less ATP productive initial glycolytic steps.
That suggests the PPP, maybe specifically this ER-associated H6PD version, could be a much more significant route for glucose breakdown and energy production than we thought.
It really does. The paper cites experimental evidence, like studies with 2DG uptake in neuronal cultures, supporting H6PD's role, and other studies where silencing H6PD in cancer cells reduced their consumption of glucose and FDG, which is a related tracer used in PDC scans.
And that contrasts with G6PD deficiency, right? That mainly affects red blood cells, doesn't it?
Yes, G6PD deficiency primarily causes problems in red blood cells due to lack of NADPH for oxidative stress defense. But deficiencies in H6PD are linked to more severe systemic diseases, suggesting H6PD plays a more fundamental role across different tissues.
Okay, so we have glucose potentially going heavily through the PPP, maybe via H6PD on the ER, generating NADPH, which fuels OXPHOS on the ER via transhydrogenase. That's a lot to rewire in the standard model.
It is. And it keeps going. Let's talk about that PPP recycling again.
Right, the product's feeding back.
The PPP produces fructose 6-phosphate and glyceraldehyde 3-phosphate. These are standard glycolytic intermediates. They can be converted back, essentially running glycolysis in reverse for those steps, to regenerate glucose 6-phosphate.
So the glucose 6-phosphate molecule can effectively go around the PPP cycle multiple times.
Exactly. Each turn generates CO2 and 2 molecules of NADPH. If it cycles 6 times, the entire glucose molecule is oxidized to 6 CO2, and you generate a whopping 12 NADPH molecules.
12 NADPH per glucose, compared to just 2 from standard glycolysis feeding the mitochondrial Krebs cycle.
A huge difference in reducing power output. And this recycling seems particularly active in tissues like the brain, which has high energy demands but also needs lots of NADPH. It could also explain some confusing results from radioactive tracer studies.
How so?
Well, if glucose carbons just went down a linear path, you could track where they end up. But if they're constantly being recycled and shuffled through the PPP and back into glycolysis, the positions of labeled carbons get randomized, making it harder to interpret the data using simple linear models. This recycling provides a potential explanation for those discrepancies.
Makes sense. So PPP recycling boosts NADPH production significantly, which then powers this extra-mitochondrial ATP synthesis.
That's the connection the paper draws. The transhydrogenase on the ER is the key link, converting that abundant PPP-derived NADPH into NADH right next to the ER's OXPHOS machinery.
What about biosynthesis then? We said glycolysis provides precursors, but the PPP also makes things, right? Like ribose for nucleotides. And NADPH is crucial for making fatty acids.
Absolutely, and this model offers a potentially simpler view of fatty acid synthesis too. The traditional view involves citrate being exported from mitochondria to provide acetyl-CoA in the cytoplasm. But if the PPP is the main route for glucose breakdown, it naturally produces intermediates that can be converted to acetyl-CoA in the cytoplasm. And critically, it produces the vast amounts of NADPH needed to actually build the fatty acid chains.
So the PPP could be providing both the building blocks, acetyl-CoA precursors, and the reducing power NADPH for fatty acid synthesis, perhaps more directly than the mitochondrial export route.
That's the implication. The paper even does a mass balance calculation showing how glucose going through the PPP cycle can neatly provide exactly the right amount of NADPH needed to synthesize palmitoyl-CoA, a common fatty acid.
And this fits with the idea that pyruvate might not primarily enter the mitochondria from glucose, right? Supporting the lactate endpoint for glycolysis.
It does. If pyruvate isn't the main fuel entering mitochondria from glucose, its conversion to lactate makes more sense as a major fate. Interestingly, there's also evidence for Krebs cycle enzymes existing in the cytosol, not just mitochondria.
Cytosolic Krebs cycle enzymes. What would they be doing?
Potentially handling acetyl-CoA generated from fatty acid breakdown, beta-oxidation, in the cytosol or peroxisomes, or maybe even from that PPP-derived acetyl-CoA pathway. It suggests metabolic pathways might be less compartmentalized than we thought. There's even evidence of lactate inside mitochondria sometimes.
Okay, this is really challenging the textbook view of compartmentalization. So let's reconsider the mitochondria overall. If they aren't the primary ATP generators from glucose and OXPHOS machinery is being exported, what is their main job in this model?
The paper reframes their primary role as focused on 1. Beta-oxidation of fatty acids to produce acetyl-CoA, and 2. Assembling and then exporting the OXPHOS machinery via these mitovesicles or through MAM contacts.
So they're like factories building the power generators and shipping them out to where the energy is needed locally.
That's a good analogy, yes.
Yeah.
Shipping them mainly to the ER, where they can be fueled by the PPP's NADPH output via transhydrogenase to make ATP for cytoplasmic processes like protein synthesis and lipid synthesis.
Is there evidence for this export beyond the vesicles? Like, do cells with high energy needs but few mitochondria show signs of this?
Yes, the paper points to plasma cells, for example. They secrete vast amounts of antibodies, which requires enormous amounts of ATP, yet they typically have relatively few mitochondria. The hypothesis is they rely heavily on this ER-based OXPHOS system.
That makes sense.
There's also evidence for mitochondrial proteins, including ATP synthase itself, being found on the outer plasma membrane of some cells.
Okay, so this revised model PPP as primary glucose route, extra-mitochondrial OXPHOS on the ER fueled by PPP NADPH, mitochondria focusing on fat breakdown and exporting OXPHOS parts. How does this change our understanding of something like cancer metabolism, the Warburg effect?
The Warburg effect. That's the observation that cancer cells consume large amounts of glucose and convert most of it to lactate, even when oxygen is plentiful, so-called aerobic glycolysis.
Right. The traditional explanation often involves defective mitochondria or a switch away from mitochondrial respiration.
But as we discussed, the ATP yield from just converting glucose to lactate via standard glycolysis is actually very low, potentially even zero or negative. It doesn't seem sufficient to power rapid cancer growth and proliferation.
So the Warburg effect needs a different explanation within this new framework.
Exactly. And this model offers one. Increased PPP activity is already a well-known hallmark of many tumors. Enzymes like transketolase, part of the PPP, are often highly expressed and linked to cancer progression.
So cancer cells might be ramping up the PPP.
That's the idea. The hypothesis is that the high energy and biosynthetic demands of cancer are met by enhancing this whole system, upregulating the PPP to generate lots of NADPH and precursors, and increasing the transfer of OXPHOS machinery from mitochondria to the ER to boost ERXPHOS ATP production.
And that would naturally lead to high glucose uptake and lactate production, fitting the Warburg observation, but for a different reason, lactate being the natural end product of this PPP-centric flow.
Potentially, yes. It views aerobic glycolysis not as defective respiration, but as a consequence of relying heavily on this PPP-ER-OXPHOS axis.
Does this have other consequences for the cancer cell? What about the mitochondria?
Well, if mitochondria are busy exporting their OXPHOS machinery, they might become less efficient at their other main job, beta-oxidation of fatty acids.
Ah. And tumors are often observed to accumulate lipids, aren't they?
Yes, lipid droplet accumulation is common in many cancers. This model provides a potential reason why. The mitochondria are functionally repurposed for exporting OXPHOS components, leaving less capacity for burning fats.
And the upregulated PTP provides both the NADPH and the Acetyl-CoA precursors needed for the cancer cells to synthesize their own fats.
Correct. So you see this functional shift. Mitochondria focus on OXPHOS export. The PPP ramps up, providing energy via ER-OXPHOS and building blocks, leading to lactate production and lipid synthesis accumulation.
What about things like circulating tumor cells or metastasis? There's evidence they actually have increased OXPHOS activity. How does that fit?
That could potentially be explained by this ER-RSKHO system as well. Maybe circulating tumor cells or metastatic sites rely heavily on this easily deployable, potentially rapidly inducible ER-based ATP production system. There's also interesting links emerging between high extracellular ATP levels near tumors, platelet activity, and metastasis, which might connect to ATP being synthesized and potentially released via extra-mitochondrial pathways.
Okay, let's try to summarize this new model then. It's quite a shift.
It is. The core idea is the pentose phosphate pathway, perhaps via H6PD on the ER, becomes the primary route for glucose breakdown, especially in cells with high energy and anabolic needs, like cancer cells.
Generating lots of NADPH and precursors.
Right. Mitochondria primarily handle fatty acid beta-oxidation and act as assembly export hubs for OXPHOS machinery,
which gets shipped maybe via vesicles or direct contact, mainly to the ER.
Where ER OXPHOS, fueled by that PPP-derived NADPH converted to NADH by transhydrogenase, generates ATP locally for cytoplasmic processes like fatty acid synthesis.
And PPT recycling enhances the NADPH yield dramatically. This whole system can largely bypass traditional glycolysis with its questionable ATP yield, and maybe even the primary use of the mitochondrial Krebs cycle for sugar metabolism.
Yes, though a cytosolic Krebs cycle might still be important, perhaps for handling acetyl-CoA from fat breakdown. And aerobic glycolysis, or the Warburg effect, might be better understood as aerobic pentose phosphate pathway metabolism.
So the main takeaway here is pretty profound. The standard model of cellular energy metabolism, the one we've relied on for so long, might really need a significant update.
It seems that way, based on this research.
The PPP and this extra-mitochondrial OXPHOS, particularly on the ER, seem poised to play much more central roles than we ever appreciated.
This new understanding, if it holds up, could resolve some long-standing scientific debates and, maybe more importantly, offer fresh perspectives on cell biology and potential medical applications.
Especially in diagnosing and maybe even treating diseases like cancer, where metabolism is so key.
Absolutely. It forces us to reconsider the fundamental division of labor within the cell and the really intricate interplay between different metabolic pathways.
It really does make you wonder, doesn't it? Could our fundamental understanding of how our cells power themselves be genuinely on the verge of a major paradigm shift?
It's certainly provocative.
And what other biological processes might be unexpectedly linked to these alternative energy pathways? That's definitely a profound question to mull over.
Indeed it is. Lots to think about.
As always, don't forget to like the video and subscribe if you haven't joined us yet.